Myself, Dr. P. Shivakumar, working as Associate Professor Biotechnology in Dr. M.S. Swaminathan Agriculture College and Research Institute, Ichangotai, Tanjavur. It is one of the Constituent College of Tamil Nadu Agriculture University. Today, I am going to talk about the genome organization, that is prokaryote versus eukaryotes. Before going to this topic, we must know some basic information about the genome, genome size, and how the genome is used for different function. The first one is the genome. The term genome is coined in 1920 by the Hans Wingler from University of Hamburg and Germany. What is the genome or defined genome? The total complement of the gene in an organism or cell is known as genome. The genome is the full complement of the genetic information in a cell and contains the program required for that cell to function. What is genome size? It refers to the total amount of DNA contained in a haploid genome expressed either in terms of the number of base pair or BB or kilobases. Coming to the genome, the largest genomes belongs to a very small creature, that is amoeba dubia. This protozoan genome has 670 billion base pairs, which is 200 times larger than the human genome. The Japan flower plant is called Paris japonica. It has the largest known eukaryotic genome with 152.23 picograms of DNA, which is 150 billion base pairs in size. The another flowering plant is called Genilisa tuberosa, has a smaller genome size at around 61 MBB size. Coming to the elements of the genome, there are two different components are very important in the genome. One is the coding region, another one is non-coding regions. The coding region is mainly or majorly contributed by the exons, or otherwise called as a coding genes, and other regulatory elements. The non-coding region is the second part of the genome is called as non-coding region. It contains, there are 4 or 5 and other components. The non-coding regions mainly concentrate the introns. The introns is otherwise called as intervening sequences. The second one is the CPG islands, is the DNA region. It contains higher concentration of C and G. Third one is the repeated sequences. The repeated sequences are normally repeated throughout the genome of the organisms. Fourth one is the transposon, or otherwise called as mobile elements. And others is retrotransposons. So these are all the two important components or elements in the genome of any organism. The slide shows the hierarchy of the gene organization from start with the smallest unit, gene, and the largest unit of the genetic material is called as genome. Gene is a single unit of genetic function. Operon is the genes transcribed in single transcript. Regulon is the gene controlled by the same regulator. Modulon, the genes modulated by the same stimulus. Element is plasmid, chromosome, page, and finally chromosome. This is the order of ascending complexity of gene to genome. The genome of all living organisms represent the hereditary material, that is called as genetic material, and is formed of DNA. Coming to the genetic materials or the organism of prokaryotes and eukaryotes, first we have to see the prokaryotic cells. The genomic DNA forms a single circular chromosome without the basic protein. It is called as histone. So in eukaryotes, it doesn't have any basic protein, or the DNA is not associated with any basic protein is called as histones. It lies in the cell cytoplasm in the nucleoid regions. Coming to the eukaryotic cell, the DNA is associated with the basic protein is called as histone, or otherwise the protein is normally associated with the DNA in the eukaryotic cells. It forms long chromatin fibers. The chromatin fibers form a network enclosed a double-layered nuclear envelope condenses into the chromosome during cell division. So the below pictures show what are the organelles present in the prokaryotic and eukaryotic cells. Coming to the genome organization, before going to talk about the genome organization, the prokaryotes, we must know the definition of the genome organization. The genome organization is nothing but linear order of DNA elements, their division into chromosome and the 3D structure of the chromosomes. The position of the DNA sequences within the nucleus and the sequential arrangement of the genes within an organism. Genome organization is depend upon the genus and is one of the main characteristics that distinguish the genera from each other. So genome organization is mainly used to differentiate the different genera or species. Genome organization, why the genome organization is very important? The genome organization is very important for the proper development and the functioning of higher organisms and is closely linked to the gene expression. That is called as a protein formation. Coming to the genome organization in prokaryotes and eukaryotes, first we can see the prokaryotes. In prokaryotes, the most genome is coding, or otherwise it contains only the exons or coding sequences. It also has small amount of non-coding sequences and regulatory sequences. Coming to the eukaryotic genome organization, eukaryotic gene cells, eukaryotic cells, mostly they have the non-coding sequence. It is around 90%. And eukaryotic cells genome also having the regulatory sequences, the introns are otherwise called as intervening sequences. It can be repeated in between the exons. So introns and exons are repeatedly present in the entire eukaryotic chromosome. The final one is the repetitive DNA. Repetitive DNA is the molecule repeatedly present throughout the genome of eukaryotic cells. What are the similarities between the genetic material of the prokaryotes and the eukaryotes? So the genetic material of prokaryotes and eukaryotes are made up of DNA molecule. So either the prokaryotes or eukaryotes, the genetic material is common, that is the DNA. They contain double-stranded DNA built by for 4 nucleotides like A, T, G, or C. Both the type of genetic material contain genes. Coming to the prokaryotic genome organization, the prokaryotic genome may be classified into 3 stages. One is nucleoid, second one is superkylling, third one is operon. What is nucleoid? Or what is the definition of the nucleoid? Genome in organized structure is called as nucleoid in prokaryotes. What is DNA superkylling? It is refers to over or underwinding of the DNA strand. Third one is operon. Operon is nothing but it contains or cluster of the genes that transcribe together to give the single messenger RNA, which therefore encodes the multiple protein. So operon here in prokaryote, the prokaryote having the polycystonic units. Coming to the prokaryotic genome organization, the prokaryotes have the smaller in size compared with the eukaryotic genome. It contains only one chromosome composed of DNA, RNA, and other components like protein. It has haploid circular genomes, that is a single set of chromosomes in an organism or in cells. The prokaryotic genomes range from about 500 kb to about 20 megabases. Generally, in the prokaryotes, the reproduction is occur mainly through asexual means. That is, mostly they follow the binary fissions. The transcription and translation take place in the same compartment. It is not in case of eukaryotes. The eukaryotes, the transcription translation occur in the two different places like nucleus and the cytoplasm. Now we can see the nuclei. What is nuclei? Nuclei is a irregular shape. Nuclei is located directly in the cytoplasm, it allowing the DNA to be contact with the cell membrane. Nuclei lacks nuclear membrane in case of prokaryotes, or otherwise it doesn't have the well-developed nuclear envelope or protective membrane. So the picture shows the nuclei of eukaryotes, prokaryotes. So the nucleotide protein helps to maintain the supercoiled structure of the DNA. Nucleoplasm, nucleosome are not found in the nuclei. It is normally present in the eukaryotic cells. Prokaryotic cells do not contain nuclei or other membrane-bound organelles. In case of eukaryotes, the membrane-bound organelles are present. Apart from the single chromosome, it contains extra chromosomal DNA. The extra chromosomal DNA is called as plasmid. Example is most of the bacteria having the extra chromosomal DNA, that is the plasmid. So coming to the supercoiling, it is important for packaging of the DNA. So this is mainly two important structures. One is toroid, that is anchoring protein. The second one is the plectronome, that is a loop domain. In the right side picture shows how the looped domain attached with histone-like anchoring proteins. So the supercoiling of the DNA to be generated and controlled by the two important enzymes. One is called as DNA kyrase, another one is called as DNA topomerase. So these enzymes are maintained in the supercoiled state of the DNA molecule of prokaryotes. What is the function of the supercoiling? It can reduce the space required for DNA packaging, it allowing for more efficient storage of the DNA molecule. Among the different type of coiling, plectonomic supercoils are predominant in prokaryotic cells because it has the circular chromosome and relatively small amount of genetic materials. Supercoiling is very important in a number of biological processes such as compacting the DNA molecule. That is called as packaging of DNA molecules. This picture shows the prokaryotic genomes organization. The first one is the prokaryotic genome in the relaxed state, the prokaryotic chromosome, that is a relaxed circular chromosome. The second one is the chromosome, circular chromosome in the supercoiled form. So that coming to the third point, third step component is operon. The prokaryotic genomes are organized into polycystonic operons or cluster of more than one coding region attached to a single promoter. The regulatory elements that control and coordinate the protein synthesis based on the cell requirement. The below picture shows how the capsid promote the RNA polymerase to bind on a promoter region, and then how the operator to enhance or block the RNA polymerase for production of or synthesis of lag operon. It contains three genes. It's called lag Z, Y, A. These three genes are called as structural genes. This slide shows the chromosome organization in the prokaryote. The first one is the DNA duplex, the folding this RNA, and the final one is called as supercoil DNA. Coming to the eukaryotic genome organization, eukaryotic genome is a multilinear chromosome. It has a multilinear chromosome and it conforms the Watson-Crick double helical structure model. So mostly the eukaryotic genome follow the Watson-Crick model of DNA. It has monocystonic transcriptional unit, but in eukaryotes, it has polycystonic units. It has mostly a discontinuous coding regions. Discontinuous coding regions, it contain both intron, exon, and exons. Mostly, introns are dominated in the eukaryotic genome. It is normally 98%. The other remaining portions, we have the exons and other regulatory elements. It has a large amount of non-coding DNA. The non-coding DNA is otherwise called as intron or intervening sequences. Eukaryotic genomes are diploid and all eukaryotic cells have two or more chromosomes. Two or more chromosomes. Eukaryotic genomes vary dramatically in terms of size and gene counts. Genome size and the number of the gene present in an organism reveal little about the organism complexity. When compared with the prokaryotic cells, eukaryotic cell having large amount of DNA in its nucleus, because nucleus is the region only present in the eukaryotic cell. It is not having the well-developed nucleus or well-developed nuclear envelope in case of prokaryotic cells. So genome in the form of nucleoprotein complex called chromatin. Coming to the what are the function of the chromatin? We can see one by one. The main function is to package the DNA into the smaller volume to fit in the cells. So the chromatin is normally involved for packaging of the DNA or DNA packaging process. The second function is to strengthen the DNA to allow mitosis and meiosis. The final function of the chromatin is to serve as a mechanism to control gene expression and DNA replication. So the below picture shows the presence of chromosome, chromatin, and DNA helix. The major protein involved in the chromatin are histone proteins. The histone proteins are otherwise called as basic proteins. So normally in eukaryotic cells, the genome may be joined together with the basic protein is called as histones. So there are variety of the RNA genes, that is a ribosomal RNA, transfer RNA, smaller nuclear RNA, etc. are present in the eukaryotic genome. It is often diploid genomes. Already we discussed it is often diploid genome. It is normally follow the sexual reproduction is a universal feature of the eukaryotic organism. Here another coming to the next, the transcription and translation take place in different compartments, like transcription occur at a nucleus, the translation occur at a cytoplasm. Whereas in prokaryote, the transcription and translation occur in only one place. There is no transcription, translation occur in nucleus and cytoplasm. Then the chromatin is made up of DNA, protein, RNA, and is dispersed in a nuclear matrix to form a network of chromatin thread. How the DNA may be transits to form a chromosome. Okay, so the major proteins involved in the chromatin are histone protein. The DNA is complexed with 8 histone proteins. It form a octamer. This form a complex called nucleosome. So the below picture, the left side picture shows the diagram of nucleosome in the side view. It contains DNA, then octamer of histones. It contains the 8 histone proteins, and another one is called as H1 histone in the right side. The right side picture is the top view of nucleosome molecules. Coming to the packaging in the eukaryotes, the nucleosome are linked by an additional histone protein that is called as a H1 histone to form a string of chromatosome. The chromosome is form a transit to the gene transit to the chromosome. 5 major histone classes are present which are associated with the eukaryotic genome, that is H1, H2A, H2B, H3 and H4. There are 2 molecules of each of 4 of histones, namely H2A, H2B, H3 and H4. This give rise to complex of 8 protein named as histone octamer or octamer. This histone octamer is flat and cylindrical particle of about 11 nanometer and thickness of nucleosome is 5.7 nanometer. These are the basic proteins which have the positive charge at normal pH, which facilitate the binding of negatively charged DNA molecule. A nucleosome with a H1 protein is chromatosome. This then coil to form a solenoid structure, that is a 6 chromosome per 10, which is condensed to form a fiber that is called as 30 nanometer fiber. This fiber then form the loops, that's a bubble, which are compressed and folded around a protein scaffold to form a chromatin. Chromatin will then supercoil during cell division to form a chromosome that are visible by using different staining method under the microscope. So the following pictures shows how the DNA to nucleosome, nucleosome to chromatosome, then solenoid, 30 nanometer fibers, chromatin, finally the chromosomes. Coming to the difference, what are the difference between the prokaryotic and the eukaryotic genome? It has much difference occur in both prokaryote as well as the eukaryotic genome. Coming to the prokaryotic genome, already we discussed many prokaryotes contain single circular chromosomes. Many prokaryotes contain a single circular chromosome, whereas in eukaryotic chromosomes, eukaryotes contain multiple linear chromosomes. So prokaryotic chromosomes are condensed in the nucleide via DNA supercoiling. So DNA supercoiling is highly helpful to condense the DNA in the nucleide. The process is called as supercoiling. And then the binding of the various architectural proteins are happening. Whereas in eukaryotic chromosomes, the eukaryotic chromosomes are condensed in a membrane-bound nucleus via histone because it has very well-developed nucleus as well as well-developed nuclear envelope or membrane. Then third one is the prokaryotic DNA can interact with the cytoplasm, transcription and translation occur simultaneously. Already this point we discussed in the early slides. In eukaryotes, transcription occur in the nucleus and translation occur in the cytoplasm. And coming to the other differences of the prokaryotes and eukaryotes, most prokaryotes contain only one copy of the gene, that is, they are haploid. But in case of eukaryotes, most eukaryotes contain two copies of gene, that is, they are diploid in nature. The another difference of the genome of the prokaryotes and eukaryotes, the non-essential prokaryotic genes are commonly encoded on extrachromosomal plasmids. So extrachromosomal plasmid or extrachromosomal genetic material are present in most of the prokaryotes. Some eukaryotic genomes are organized into the operon, but most are not in eukaryotic genome. The final one is the prokaryotic genomes are efficient and compact, and it contains only the little repeating DNA, but in case of eukaryotic, it have more repeated DNA sequences. Extrachromosomal plasmid are not commonly present in the eukaryotes. So these are the some important difference of the prokaryote versus eukaryote genomes. Coming to the comparative genome size of the organism, here the slide shows the important organism or the model organism for developmental genetics, molecular biology, microbiology. We know what is the genome size and what are the chromosome numbers and how many genes present in the genome. The first one is the Homo sapiens. The genome size is 3.2 billion base pairs. It contains around 25 genes. It has 46 chromosome number. Then mouse is a model organism. So the mus musculus, the genome size is a 2.6 billion base pairs. It also contains 25,000 genes. It has 40 chromosomes. The third one is the Drosophila melanogaster, or otherwise called as a fruit fly. It is a model organism for developmental genetics. The genome size is 137 million base pairs. It contains around 13,000 genes. It has only 8 chromosomes. The important one, the model organism for plant is Arabidopsis thaliana. The genome size is 100 million base pairs. The gene number is around 25,000. It has 10 chromosome number. The C. elegans is also a model organism for nematology. The genome size is around 97.97 million base pairs. It contains around 90,000 genes. It has 12 chromosomes. Coming to the yeast, that is a Saccharomyces cerevisiae, is also a model organism for the microbiologist working in the yeast. The size of the genome is around 12.1 million base pairs. It contains around 6,000 genes. It has 32 chromosomes. Another model organism for microbiology, that is called E. coli or bacteria. The genome size is 4.6 million base pairs. It roughly contains 3,200 genes. It has only one chromosomes. The another final one and another example is Haemophilus influenza is a bacteria. The genome size is 1.8 million base pairs. The gene number is around 1,700. It also has only the one chromosomes. So this lecture, we clearly discuss about differentiate how the genomes are organized in the prokaryote as well as the eukaryote. So this information is highly helpful for you to easily understand of the prokaryotic and the eukaryotic cells. Thank you, one and all present here. Thank you.